Okay, it's my pleasure today to introduce our speaker, Carsten Prasse. Carsten did his I've known Carsten for a while now because we travel in the same conference circles, right? And so he did his bachelor's and master's degree at the University of Beirut, and then his PhD at Koblenz Landau, and then stayed there for a postdoc for a little while. And for those of you in the emerging contaminant area, he worked with Thomas Turnus, who's known in that area. And then he did a postdoc at UC Berkeley with David Sedlak. And then he moved to Johns Hopkins University in the Environmental Health and Engineering department, which makes him essentially my academic stepdad, because he took essentially the position that my PhD advisor used to have. So my advisor retired. So even though I'm just a little bit older than Carsten is. So thanks for being here, and look forward to your talk.
Thank you so much. Interesting to think about myself as your stepdad. Okay, I might have to think about this for a bit. Okay, I hope, let's see. I hope everybody online can hear me. Thank you, Bill, for the introduction, and thank you all for coming. And it's a pleasure to be here. I told Bill my wife is 36 weeks pregnant, so this is actually my last seminar trip that I'm going to be doing for a while. So it's great to be here, and also great that the little one didn't decide to come out early. At least, based on my phone, it hasn't happened yet. So I hope it will stay that way until at least tomorrow. And yeah, so I'm in environmental health and engineering, and I'm going to be talking today about one of the main focus areas that we've been working on, that I've been interested since my PhD, which is chemical exposures, and especially chemical exposures when we think about complex mixtures and how we deal with them. Both from the assessment side, but also from the engineering side, and how we treat or how we design treatment systems, for example. So the first, I know it's still relatively early, so kind of like a question for you all. How many chemicals do you think we put willingly every day on our bodies? Again, the reason why I'm asking this, just to give you a bit of a sense why I've been really thinking about this a bit more. So please shout out 20?
50 for women and 25 for men.
All right. Any other numbers?
2,000. That's what practice that I have.
350. All right. So, but it's actually interesting with the surfactants and everything. So there was a study by the Environmental Working Group that came out a few years ago that did this, and they estimated it's about 168 chemicals for women and roughly half of that for men. But obviously there are so many more chemicals that we know about now. So the number, I'm assuming, actually has probably increased when we think about all the other compounds that might be present in, for example, personal care products. So this is obviously a relatively high number, but I think when we think about all the chemicals that are in commercial use, the numbers get even higher. And so when we think about detergents and cleaning agents that we're using in our houses, and one of the things that I'm really thinking a lot about is like, okay, if we're using them in our houses and then they go down the drain, what happens to them? And what are the impacts of that? And what we've seen, and I kind of gonna ask this somewhat provocative questions, is, are we looking for the right compounds? And I hope over the course of my talk it will become clear what I mean by this. But what we've seen over the last year, which is kind of like illustrated here a bit, is that based on this high use of chemicals and increasing use of chemicals, we've seen this dramatic increase of the chemicals that we can detect in the environment. But it's not only because we use more chemicals, but also because we use more and more sophisticated analytical tools. Like my lab, for example, we use high-resolution mass spectrometry that allow us to detect thousands of chemicals in a single sample today. And then we also have an increased sensitivity of analytical instruments. So we have a lot of chemicals that we know are present in the environment. And then another aspect, and that's especially relevant for when we think about engineering, is that we have a lot of treatment approaches. I mean, we are primarily working on water, where we form a lot of transformation products. So typically, if we degrade a contaminant, they generally don't just disappear, but they form transformation products. So this number that I showed you earlier is likely to be in the hundreds of thousands when we also include these transformation products. Now, what's interesting when we think about how we assess these transformation products and the formation of these, and that's something that I've also done. Typically, we do lab experiments where we investigate a single compound, like illustrated here in purple, going down, and then you see different transformation products forming. And typically what we do is we focus on those that are most abundant, which is because they're typically most easy to detect. But the point I want to make is that obviously just because they're most abundant doesn't mean that they're the most problematic ones. But it could also be that one of these compounds that are formed in much lower yields is actually more toxicologically relevant. And I will be coming back to this in a bit. And then the other thing I think that we always have to keep in mind is that despite these advances of analytical tools that we're using today, and I'm showing you here like analytical windows for the technology that we are using, which is liquid chromatography, mass spectrometry. All these tools have only certain analytical windows. So we will never, I believe we will never be able to detect all compounds that are present in, for example, a water sample with a single analytical method. So we are, with each method, we can only detect a certain analytical window. And that's important when we think about certain compounds that are present in the environment or that might be formed in the environment. And one example I want to give you that might be hopefully familiar to most of you are disinfection byproducts that we drink. Most of us drink on a daily basis because they are present in our drinking water. And it's interesting that we've done more than 50 years of research on these disinfection byproducts, and we know more than 700 today. But we still only know about 40% of the actual organic halogen content that's present in disinfected water. And the question that I kind of want to ask is, and that's coming back to the question that I raised in the beginning, why is that? And again, could it be because we might not use the right analytical tools, or we are not able to detect all the compounds that we actually want to look for with the tools we are currently using? And this is important, not just because we want to close this mass balance which is interesting, obviously, from a scientific standpoint, but it's also important to think about, like, with the disinfection byproducts we know, we actually only can explain a small fraction of the toxicity that we detect in water. So this is taken from a study that was published in 2020 by Daniel Stalter, who basically tried to explain with the known DBPs how much of in tap water, the tap water toxicity can be explained. And what you can see is this is a log scale. Typically, we are at lower than 10%. So 90% of the toxicity actually we can't explain. And again, emphasizing that we need to know more about disinfection byproducts that are present and that are causing also this toxicity. And then one important aspect from the methodological side is also that often with these contaminants, we're talking about contaminants that are present at very low concentrations. And so typically what we have to do is we need a pre-concentration step. So for example, using solid phase extraction. And that's necessary for some of the chemical analysis, but it's especially also often necessary for the ecotoxicological evaluation when we think, for example, about like in vitro bioassays. The issue with that is, though, and this is a very simple experiment that I did a few years ago, what we often do is we use solid phase extraction to concentrate basically the organic compounds that are present. And so what I'm showing you here is just a simple result where I, in this case, wastewater, ozonated wastewater before and after, and I measured the DOC. And then I extracted that water with different commonly used SPE cartridges. And so if these sorbents would work perfectly, we wouldn't see any concentration because this is kind of measured in what's basically running through the cartridges. But what you can see is that between 20 and up to 60% of the carbon that's present is not being absorbed to these cartridges. And obviously, if you're then doing toxicological or ecotoxicological assessment on those, you won't take these compounds into account. So you only get an information about the compounds that actually stick to these adsorbents. And so this is very important because what this means is that when we assess different water samples, and obviously you can also think about other types of samples, we really restrict our assessment to those compounds that we can extract or detect. Okay? And one of the classes of compounds that I'm particularly interested in are low molecular weight and highly polar compounds. And in my opinion, they really represent a big challenge for the current analytical and ecotoxicological methods that we're using. Now you might ask, well, why do we care about these compounds? Well, when you think about in engineering how our field has advanced and the different approaches that we are increasingly using, these compounds are actually very relevant because, for example, we're using more and more oxidative water treatment systems that, for example, use UV hydrogen peroxide or that use ozone, that generate a lot of these small molecular weight compounds. Then we're also using membrane treatment, and a lot of these low molecular weight compounds, at least if they're uncharged, they might actually go through these membranes, so even reverse osmosis might not be necessarily a good barrier for them. And then we're also increasingly using other approaches like electrochemical water treatment or resource recovery, where we have a lot of like radicals. This is a study that we published when I was working with David Sedlak, where we looked into these systems, and there's a lot of radical chemistry that goes on that leads to all different kinds of transformation products. And then obviously also by having worked in California specifically, we're thinking more and more about water reuse or centralized versus decentralized system. And also there the question is, do we really have the adequate methods to assess all of these different treatment technologies? And again, with the limitations of the current approaches that we have. So what I think, and I hope I made this clear to all of you why I think this, is that we really need to rethink how we evaluate and also how we treat drinking water. And one way I've been thinking about this is, and this is kind of, for those of you who have heard of personalized medicine, this is kind of like adapted from this. What we're currently doing is this one treatment fits all, where we have different drinking water sources, and we have basically water treatment processes that are often very similar to each other. And then in most cases, we are getting drinking water that is, has a reduced toxicity that is definitely safer to drink. But we also know examples where the toxicity remains similar or even increases when you think, for example, about disinfection byproducts. So basically what I'm proposing is that with all the knowledge that we have today and also the technological advances, we can do better than this. And we can actually do what I call personalized source water treatment diagnostics and treatment. What I mean by this is that we actually now have the tools to better evaluate what's in our source water, to basically evaluate which treatment approach could be really the best one for this specific water, to then ultimately really get a reduced toxicity across the board. And if you wanna know more about this, Paul Ferraro, who's a colleague in my department, and we published a perspective piece in Nature Sustainability last year where we outlined this approach. We call it Miami, which is an acronym for Mixture, Assay, Measure, Innovate. And the main part of this is really that instead of doing individual chemical assessment, which is what we're doing right now, we really have to start with this complex mixture if we really want to target the protection of drinking water consumers. And so, but if we want to do this, one thing that is missing right now is we really need approaches that allow us to assess these complex chemical mixtures. And I've shown you these larger numbers of chemicals before. Obviously, you might ask, well, with so many chemicals, there isn't really a way of assessing every single one of them, and I would fully agree with that. So what I'm proposing is basically we kind of have to think about a prioritization. So we don't have to, when we think about just transformation products, we don't really have to think necessarily about compounds that are mineralized or that are non-toxic, but we really have to kind of find ways to focus on those toxic compounds that are of health concern. The issue, though, is that when we think about the analytical chemistry on the one side and the toxicology on the other side, is that they are typically disjoint. So we don't really have a lot of methods that actually combine these two approaches. There's one main exemption that I want to briefly mention, which is that some of you might have heard of, which is called effect-directed analysis. What this does is it takes an environmental sample, it puts it through a biotesting, and then if there's an effect, it fractionates the sample, and then each of the different fractions is tested. And the ultimate goal is basically to reduce the number of chemicals that are present in each fraction to then ultimately identify the relevant toxicant. The issue with that is that, similar to what I said before, this approach also relies on sample enrichment, like the solid phase extraction. And it also relies on pretty heavily on sample manipulation, so such as fractionation, where you might actually lose a substantial amount of the compounds. All right. So when I was at Berkeley, as a postdoc, I was working in the Superfund program, and I was working with toxicologists and actually audited a few classes in toxicology to learn more about how do toxicologists actually think about this. And when you think about the classical toxicological approaches, which is kind of like illustrated here, where you have an exposure, so you expose, for example, a fish to a chemical, and then you measure an adverse outcome. So for example, the fish dies. This was the classical approach, which is kind of like a black box. We don't really know necessarily what's happening in between, like why specifically is the fish dying. In toxicology, over the last 10 to 20 years, this has led to the development of a different concept, which is called adverse outcome pathway concept. One of the reasons also for this development is that we have more and more tools available. A lot of this is also based on omics, like proteomics and metabolomics. And what this adverse outcome pathway basically does is it talks more about, okay, what are the exact mechanism? And one of the things that I want to focus on is this molecular initiating event. But maybe to give you a general sense what this adverse outcome pathway concept is. So we have a so-called molecular initiating event where you have an interaction of a chemical with a biomolecule that can be either the covalent binding to proteins or DNA, or it can be binding to a receptor, which when you think about endocrine disrupting chemicals, for example, is relevant. And this leads to a variety of different key events that then ultimately lead to adverse outcomes. When I learned about this first, I was really intrigued by this first part, and especially the protein binding and the DNA binding. And once I started thinking about this more, when you think about chemicals that actually bind, so we are talking about covalent binding in this case, that react with protein and DNA. These are electrophiles, organic electrophiles that are particularly relevant for this. And among all the toxicants that we know, these electrophiles are actually the largest group of toxic chemicals that we know at this point. So they are major contributors to why chemicals are toxic. And it's interesting when we think about drinking water, these have actually also been seen in drinking water. So these are examples of electrophiles that have been identified in drinking water, so far primarily individually with very specialized approaches. But I'm just showing you this to kind of illustrate that organic electrophiles are formed also in different treatment systems. So this really got my thinking going about, is there a way of assessing the formation of these compounds more holistically? And I developed this, what I call reactivity-directed analysis approach that kind of combines the toxicity assessment with the contaminant identification. I published this critical review paper last year, if you want to read more about it. It kind of like outlines a bit my thinking about how this has already been used in the past or similar, and how we can use it, especially in the environmental field. One thing I want to mention is that just for kind of that we're all on the same page, you probably have all heard of in vivo assays, toxicity assays, which basically means you use animals. Most of you probably also have heard of in vitro, which means you're using cell. And the approaches we are using are called in chemical. So in this case, we are not using organisms, or we're not using cells, but we're actually just using biomolecules, such as proteins or, for example, DNA. We've so far primarily focused on proteins or amino acids. And we are particularly interested in these two amino acids that we all have in our bodies as part of different proteins, because we already know that these are the main reactive sites for electrophiles that we are exposed to. So cysteine is one which has this thiol group here, and then lysine is another one that has this primary amine group here at the end. And this approach that I'm talking about actually has already been used to some extent. What I'm showing you here is an OECD guideline that assesses the skin sensitization of different chemicals. So this assay uses these peptides, and one of them has also this thiol group here from cysteine, and the other one has this primary amine group. And this is used, for example, by the cosmetics industry to evaluate if you have certain chemicals that are reactive with proteins on your skin that could lead to irritation of your skin or to allergic reactions. But this is so far only used for individual chemicals and for known chemicals. So, and we are, in our thinking, we've been thinking more about, can we actually apply this for more complex systems? And when you think about kind of, again, how we identify transformation products that are formed in different environmental systems, typically what we do is we do lab experiments, we degrade organic contaminants, and then what we typically do is we identify these transformation products with, for example, liquid chromatography, high-resolution mass spectrometry. Now, this reactivity-directed analysis approach, it kind of introduces an additional step where we're adding a nucleophilic probe. So in this case, for example, this lysine, which is one of the amino acids that I had mentioned. And this reacts specifically with these with electrophilic transformation products that are formed. And then we are basically analyzing, using this instrument to analyze the adducts or the reaction products with this amino acid. Okay, so how is this useful for us? I want to give an example of kind of how we've been using this so far, and then at the end also give an outline how we want to use this in the future. One of the things that we are working on right now, or we worked over the last years, are phenolic compounds. So phenolic compounds are very commonly used in different, a variety of different anthropogenic chemicals. So bisphenol A is probably known to all of us. Our plastic bottles now say they are bisphenol A free, but that only means they contain bisphenol F, for example, and other compounds. But in addition to all of these anthropogenic chemicals, phenols are also very relevant as natural phenols, which is kind of like indicated down here, or they're present in natural antioxidants. So they're very commonly found, for example, in our drinking water, just because they're part of natural organic matter. And the reason why we are looking into them is they have been shown to be precursors to a lot of disinfection byproducts. So we wanted to know more about what is happening, or what kind of electrophilic transformation products that are being formed. And what's also interesting is that these phenolic compounds have been studied for, at least for chlorine, for probably more than 70 years. And for hydroxyradicals, for example, we know that they are oxidized to form these polyhydroxylated species, and then ultimately they are broken down to these carboxylic acids. At least that's what we know at this point. And then for chlorine, what we think, or what we know is happening, you have these chlorophenols that are forming, and then if they react further with chlorine, you form trihalomethanes and haloacetic acids, which are the regulated disinfection byproducts that we have in drinking water today. But it's interesting that these only, in the chlorine case, these two only account for 30% of the mass balance. So again, we only know a small fraction of the compounds that are actually being formed. So there's still a big knowledge gap, and the same is actually the case up here. Okay, so I'm just going to walk you through kind of how our experiments look like. So in this case, I'm showing you oxidation of phenol with hydroxyradicals. And what you can see is what is illustrated here. You see phenol going down, and then we added this amino acid, which is shown here, and we observed this formation of this adduct with mass 255. The reason why high-resolution mass spectrometry for us is so important is because what it allows us is to determine the masses of these compounds with a very high accuracy. So we are getting, for example, we're getting 5-digit accuracy, and what this enables us is to really get the sum formula of these compounds. So basically what we're doing is we're knowing the parent, like the mass of lysine itself, and then we are looking for the adduct, and basically by subtraction, you can determine what the adduct mass is. And this took us a pretty long time to actually identify which compound is responsible, but we were able to identify this dicarbonyl compound as one as this reaction product. And this was actually the first time that this compound was detected in water. And again, one of the reasons is that this compound is very difficult to detect with the conventional analytical approaches that we have. What's interesting is, again, coming back to the yield discussion, is that for hydroxyradicals, this compound actually only has a relatively low yield of around 2%. But again, we know, or we have strong indication that it is highly toxic. And in the chlorine case, we found it in yields of 18%. One thing I want to mention is that for chlorine, we also found this chlorinated species, but we weren't able to generate a reference standard, so that's why I'm not providing you any yields here. But we saw it for hydroxyradicals, and we saw it for chlorine, and we're now also working on ozone. And also here, which is kind of like illustrated here in gold, also for ozone, we can see this dicarbonyl compound being formed. So it seems to be a very commonly transformation product of phenolic species by a variety of different oxidants, okay? And I've showed you the results for phenol, but we've now tested a whole different variety of different phenolic compounds because, again, they are very abundant in natural organic matter, in anthropogenic chemicals. And we can we found these dicarbonyl species for most of the phenols that we tested. Okay, so they are it looks like they should be present in a lot of the oxidized water samples. Okay, so one other aspect I want to mention, because again, we are only looking at the reaction of these compounds with amino acids, so obviously you would ask, well, how relevant is this for toxicity? While I was at Berkeley, I was working with Dan Nomura, who's in the toxicology department at Berkeley. He uses an approach that is called chemoproteomics. I don't want to go into detail, but basically what this approach does is it allows you to assess the reaction of a chemical with a whole proteome. So in this case, we looked at mouse liver proteome, and specifically at cysteine. And what I'm showing you here is that if you add this dicarbonyl compound, it reacts with a whole variety of different proteins. Okay. So we were able to demonstrate that it is highly reactive, and it is able to react with a lot of different proteins. And one of the things we did for one of the proteins that we saw reactivity, which is GAPTH, which is an enzyme that's in all of our bodies that's involved for the breakdown of glucose to obtain energy, we basically did a simple or conventional enzyme activity assay, and you can see that when you add BDA, we have a substantial reduction of the enzyme activity. So if you would be exposed to this compound, or if this enzyme would be exposed to this compound, you would definitely see an impact on its activity. The other aspect that was very interesting is that this compound actually had been seen before, not in water, but actually in our body. If you're exposed to furan, and furan is present, for example, in wood smoke or in cigarette smoke, what happens in your liver by cytochrome P450 enzymes, they're actually transforming furan to the same compound. And that was interesting for us because there's actually a whole variety of different studies that show that this compound also reacts with different DNA bases. And there's actually Lisa Peterson, who is actually here in Minnesota. She did a lot of work on the DNA reactivity of this compound. So again, just demonstrating that while we're using a very simplified system to detect this compound, we definitely see that it is able to identify compounds that are highly toxic. Okay, I'm an environmental chemist, so in addition to kind of like the assessment of the toxicity, we are also very interested in what are the formation mechanisms of these compounds. And for those of you who have worked with chlorine, and I'm just maybe to steal my own thunder, we still haven't figured out the story entirely, but it is very interesting that we start with this C6 compound and we chlorinate it, and then we end up with a C4 compound that doesn't have any chlorine, which is kind of very counterintuitive. And again, we're still trying to solve the puzzle. But among the things we did is, the first thing we tried is like, okay, if it's happening with phenol, and I've showed before that chlorophenols are the next compounds if you chlorinate phenol. So we did experiments with a variety of different phenols, and we were actually able to identify that these BDA and chloro-BDA are also formed from a lot of the phenolic compounds that we see. So this was to confirm that BDA and chloro-BDA are formed from this pathway that we know, that phenol reacting to chlorophenols, mono and di and trichlorophenol, and then reacting to BDA. So that's a confirmation. But at least based on literature now, what has been proposed is that if you add more chlorine to trichlorophenol, chlorine reacts at this para position here to the hydroxyl group, and you form this benzoquinone. But when we actually did experiments with this benzoquinone, we didn't see any formation of BDA and chloro-BDA. So again, that was for us an indication that there must be a different pathway that's relevant for this. One thing that helped us here is one of the model compounds that we looked at, which is this catechol compound. And that was actually our record breaker in terms of BDA formation, because for this compound, we actually saw that more than 40% is transformed into BDA, okay? And so what we think is that actually this catechol here is critical for the formation or in the formation mechanism. So our thinking right now is that, well, I showed before that the chlorine reacted at this para position, but what we actually think, well, maybe it's reacting at the ortho position and then ultimately leading to this orthoquinone species that then results in the formation of BDA and chloro-BDA. Unfortunately, we haven't been able to just purchase the quinone, but the catechol itself should be oxidized fairly quickly to this compound. So we have pretty good evidence that this is the intermediate in our reaction, and we actually were able to identify this compound in chlorination experiments with phenols. The other thing is kind of to look more into this. Well, again, we have a C6 compound, and we end up with a C4 compound. So one of the questions that we've been asking is, well, which of these carbons are actually ending up in the C4 compound? And to do this, we actually are working with these three gentlemen who were working on a different project on trihalomethane formation from phenols. And Keith Reber, who is a synthetic chemist at Towson University, and so John Sivvy is also at Towson University, and Dan McCurry is at University of Southern California. But Keith Reber, he synthesized these stable isotope labeled compounds. So you can't buy them, but he's a very good synthetic chemist, so he synthesized these. So we've been using these now to use the carbon label, the stable isotope labeling, to identify which of the carbons are actually ending up in our C4 compound. And what we were able to show is by basically doing chlorination experiments with all these different compounds and then comparing like which of the stable isotope signatures actually is still present in our C4 compound, we were actually able to identify that these four carbons are the ones that are responsible or that are present in our C4 compound. Okay. Unfortunately, at this point, at least, I'm ending the story here. As I said, we're still trying to figure this out. I think we're pretty close. But one main question is still, again, with one puzzle piece we haven't figured out is kind of where the chlorine is going, basically, in that whole reaction pathway. But again, I hope maybe within the next year I will be able to answer that question. It's been an interesting journey to solve this puzzle. All right, but taking a step back, one thing I guess wanna just summarize is that I hope I will show you that this new approach, reactivity-directed analysis, really allows us to assess the formation of electrophiles and also identify basically what they are, because for these dicarbonate species, as I mentioned, we haven't been able to identify them before in water. I think one of the promises that we see is really that we have also a measure for toxicity with this approach. So it combines the chemical identification with the toxicity assessment. And it might be really a useful tool to close this gap that we have right now for these low molecular weight compounds that are so challenging to detect with the other analytical approaches that we have. Where are we taking this? And this is actually, I was awarded luckily this year an NSF Career Award, where we're going to be able to continue this work. One of the things that we are now thinking about, I showed you these lab experiments with individual compounds, which is all nice and fine. But obviously, we're using much higher concentrations than we typically find in the environment. And one thing that we've been thinking of is like similar to like I introduced solid phase extraction, using a modified approach that we call solid phase reactivity-directed extraction, where we basically have microbeads that are surface functionalized with these nucleophilic groups that we can then use either in a fluorescence assay, where you basically have your control, where you don't add anything and you add a fluorescent probe at the end that just reacts with all the nucleophiles on the surface, and then you compare it with a sample where you had electrophiles present. So basically you get a reduced fluorescence signal. And I hope that the goal is that we could use this as what I call total organic electrophile assay. It just gives you an overall sense of how many electrophiles are actually present. And the other thing is we also using this now for the mass spectrometry, where we actually using an LC-MS basis. This is actually based on click chemistry, and well, as you know, the Nobel Prize in chemistry was awarded this year to click chemistry. So this is heavily influenced by the research that has been done by Karen Berlossi and others, where we basically functionalizing the beads with these nucleophiles that can then be cleaved from the surface, for example, with light, so that we can analyze these surface functional groups in our LC-MS. Okay, but this project just started. And I hope over the next few years we will definitely apply this also for real treatment samples. Okay, then the other thing maybe I also wanted to talk about is kind of where I see this approach or how this approach can be applied. I think it can really be helpful in the development and assessment of new advanced drinking water treatment approaches. So again, currently we don't really have, with the analytical tools we commonly use, we might miss a lot of the byproducts that are being formed. It also can really help us to assess the formation of toxic byproducts. So instead of just looking at all the byproducts that are being formed, really honing in on the ones that are most problematic. I don't think this approach will ever replace all the other toxicological approaches. That's not my thinking, but I think that this approach might be helpful to really help us prioritize which are the chemicals that we really need to focus on in a more in-depth toxicological evaluation. And then I've talked a lot about water, but we just submitted an R01 to NIH where we're actually going to look into using this approach for e-cigarettes. So you can think about it from the perspective of water, but e-cigarettes are another interesting area that we started working on, because despite the fact that the industry tries to tell us that e-cigarettes are a healthy alternative, we actually know that there's radicals being formed, and there is a lot of like electrophiles that are being formed. So we are implementing this right now to also look into e-cigarettes or into air in general. And then obviously, I mean, the ultimate goal would really be thinking about how does our drinking water regulation look in the future, and how can we use approaches like this and others to really prioritize other chemicals that we should really focus on. All right, with this, I want to thank my great research group. I also want to thank the different funding agencies. And then I also thank you for your attention, and I'm happy to answer any questions.
Thanks for a great talk, Carsten. I have kind of a big picture question for you. When you think about binding, it doesn't always lead to disease. And I would think that a lot of times, in fact, it won't lead to disease. It might only lead to disease for a certain very small subset of sensitive people.
Yes.
And so I just wondered if you could comment, especially this idea that you put up at the end and at the beginning of sort of tailoring our treatment or tailoring regulations to some of these findings. I wanted you to comment on kind of the balance, the pros and cons of protecting this sensitive population that may be affected versus maybe doing a lot of treatment that is resulting in huge greenhouse gas emissions, electricity use that might not be necessary. So kind of wanted you to comment on that.
Well, actually, yeah, thank you for thank you for the question. And actually, before maybe one thing I want to mention is that the last part, what you just mentioned in terms of the treatment aspect, I actually think we should treat less. And just because, I mean, well, I'm from Europe, so we don't really, Germany, we chlorinate to some extent, but not really. In the US, we just chlorinate across the board. And we know that disinfection byproducts are a major driver of toxicity and we know there is a clear association with bladder cancer. So I'm actually not thinking that we should treat more. I think we should treat more selectively. I am not saying chlorination is bad. It has saved billions of lives and it will save billions of lives, but I don't think it's necessarily everywhere. So, and I actually have an issue a bit with, and again, having been living in California with water reuse, like exactly what you mentioned. The treatment trains we're using right now, it's ridiculous in terms of like the different stages, the energy consumption, and then also again, like thinking about how do we assess this, because we're forming all these different byproducts. So maybe just upfront, I actually think we should treat less or more. I mean, that's kind of why I use this personalized treatment approach, because I think, so that also comes into this. Then the other aspect that you mentioned, and actually, well, everybody, we talk a lot about PFAS these days. And I think PFAS is a great, I mean, with my approach also, you won't be able to detect PFAS because PFAS doesn't covalently bind. But the reason why I bring up PFAS is, so we just published a pilot study that was set here in Minnesota where we looked into groundwater treatment with activated carbon and NI exchange resins. And when we talked to, well, utilities and also health officials, they've known about PFAS for more than 20 years in groundwater. They've known that people are exposed to it, but they've done nothing. And they've done nothing because it hasn't been regulated. But my point is, does it really have to take 20 years for us to get to the point where we regulate things. And we still struggle with this. And now with EPA having these extremely low concentration or recommended levels, obviously that has led to a whole different issue about like how are we dealing with this. But my main frustration really with why I've been thinking a lot about this is kind of, I don't think our drinking water regulation is really necessarily geared to protect us. Because again, it is a very, it is not a proactive system. It's like, it's very much, it's reactive to something that we have. And for disinfection byproducts, we actually know that the regulated ones that were originally considered as being basically sentinels or indicators, they actually aren't good indicators. So also there, we don't really think about the protection of the people. And the other issue is also that the drinking water industry has, because of these byproducts being regulated, shifted from chlorination to chloramination, where these are formed in much lower concentrations. But now we know that there are all these nitrogenous DGPs formed that are much more toxic. We know that already. So again, so that's just, sorry, that was kind of a long introduction, I guess, but this is kind of like exactly why I've been thinking about these things. And now to the main question that you ask about, like, how do I see this to be implemented? It is very challenging, and it will definitely take a long time, also because we really have to kind of we have to rethink our regulatory system too, because right now what we regulate is individual contaminants and specific concentrations. If you're using bioassays, you won't necessarily get, you don't necessarily get a specific concentration. You might get a threshold concentration. But my thinking is that, and in Europe, for example, people have definitely thought more about this too, or implementing it already on the wastewater side, but also increasing on the drinking water side, is using a battery of different bioassays to assess different endpoints. So I think that's kind of where I would think this needs to go. But again, there is a lot of questions still about how do we exactly do this. The point that you raised with the different sensitivities, my answer would be that shouldn't keep us from doing it, to be honest, because I would rather protect the most vulnerable. I mean, when you think about, again, children, for example, I would rather have a system that protects them, and it wouldn't be necessarily as impactful for us or for me as an adult. But I think that's how I would see it happening. And in Germany, for example, in drinking water, we have this precautionary principle which exists. So in Germany, if a utility detects a compound that we don't know about, they immediately have to go and test it. They have to report it to the Environmental Protection Agency in Germany. They have to test it. And within half a year to a year, they generally have a result. So that's kind of like, again, I hope this answers your question, but it's not easy, but I think, again, I think we have to think about it, because again, with all the chemicals that are out there, we're just overwhelmed by the sheer number, and all the compounds that we drink or that we eat. We could also obviously think about other sources, but yeah.
Thank you.
Sure.
Yeah, I agree. Great talk. In regards to one of your earlier slides where you showed the disinfection byproducts only accounting for 1% of the toxicity, your fractionation kind of suffers from the same thing that the TIE did from years ago, where we're separating out these compounds based on toxicity, but then we're removing any synergy or antagonism going on. Can you speak to that? Do we know anything about those the known DBPs? Is there synergy or antagonism? And then can your method sort of tease that out, or does it not?
First of all, I would say, and Beate Escher, who is in Germany at UFZ, she's done a lot of work on mixture toxicity. We actually know that a lot of the effects we see are additive effects. So to maybe say this up front. And we are actually, so one thing we are doing right now is actually thinking about how does toxicity correlate with reactivity. So one of my PhD students, she's actually using she's doing lab experiments with individual electrophiles, but then also using computational chemistry tools to think about, can we also predict this reactivity? And there are some studies who actually show that it is very, well, first of all, on an individual compound basis, you can actually predict it relatively well. And then one thing I actually didn't mention, that Michael Pleber, who was at the University of Illinois, he's an emeritus now, but he actually developed a thiol reactivity assay, where he basically adds a thiol to water and just measures how much is reacting with the file. The issue there is that he still uses an extraction, so he doesn't use like the bead approach. But they have actually correlated with the toxicity they see in different in vitro assays. And they actually see a fairly good correlation with this. So it looks like that this approach can definitely give you a good indication about the overall toxicity of a water.
So it being additive would lend credence to what you're doing in terms of the chemo, the chemical binding.
Yes.
Yeah.
And again, I mean, yeah, so that part, and again, I mean, the reason why I originally started thinking about this is really also from the identification part, because again, just like thinking about how do we identify chemicals that are out there that are limited with the approaches we have right now. So, but yeah.
Great talk. Thanks for your presentation. And I have a question about the overall toxicity of the disinfection byproducts. Now the regulation for the disinfection byproducts is set by the individual compound, right? Mm-hmm. But if you put all the disinfection byproducts in the water and consider the net toxicity of all, I'm wondering if it will, like, if we analyze the toxicity by your method, it would kind of be more toxic than is, individual things. Have you considered doing this part?
Yes, I have considered. We haven't done it, but I can tell you there was actually just a study that was published by Susan Richardson, who has done a lot of work on disinfection byproducts, who's probably the most well-known researcher, at least in the US, in this field. And they've actually done a study where they looked into the extraction methods that we're currently using. And so they were actually able to show that basically they just spiked a whole different mixture of different disinfection byproducts and then use different extraction approaches. And they were actually able to show that for a lot of the disinfection byproducts, you lose 80 or 90%. So you lose a lot of the compounds that are present in your sample that you wouldn't take into account in your subsequent toxicity assay. And in that sense, at least if they are sufficiently reactive with our approach, you would be able to capture them. Yes, but we haven't done that comparison. Once we have this microbead approach that I show you developed, and that's something that one of my PhD students is working on right now, then we will certainly hopefully do this, where we do a one-to-one comparison basically of what we see, and also how it changes with different conditions. So, but yeah, it's a great question. Thank you so much. Thank you all.